Welcome dear students of St. Peter's College, Colombo and Gambur branch. Today my lesson is about molecular biology, unit 7. Today I am going to talk an important topic on molecular biology, recombinant DNA technology and DNA cloning. Recombinant DNA technology means integration of different DNA fragments of two sources, and DNA cloning gives an exact copy of an organism which is having the same genotype and also the phenotype. It means formation of an organism such as you and me, an exact copy of us. But actually human DNA cloning is not approved. Only some crop animals as well as plants are cloned. If we go to the details of this lesson, the basis of recombinant DNA technology is depending on the genetic code. All the organisms are sharing same genetic information stored in DNA, and at chemical level DNA is similar in all the organisms. Organisms share the same genetic code. It may be a bacteria or a plant cell. They share the same genetic code. So the same peptide is encoded in a particular gene, independent of the organism. In recombinant DNA technology, recombinant DNA molecules are important. So, recombinant DNA molecules are formed by new genetic combination of two organisms. It may be a target gene like insulin, it is a human gene, and it is integrated into a bacterial plasmid, and this is the recombinant plasmid, the recombinant DNA molecule. So two or more different species are joined together and inserted to a host, forming a new recombinant combination. So recombinant DNA molecules are DNA molecules that are made in the laboratory using different methods of the genetic recombination, and which brings together the DNA from different sources, creating some new sequences which are not found naturally. There are various techniques used in recombinant DNA technology, that is isolation of DNA from different sources and restriction digestion using restriction enzymes, and separation of DNA fragments using the technique agorostate electrophoresis, and joining the DNA fragments using DNA ligase enzyme, and also identification of correct fragments which is having the desired nucleotide order using marker genes or DNA probes, and inserting the DNA molecules to the host cell. Usually the most common method is transformation and also DNA cloning. Those are the techniques which are used in recombinant DNA technology, and it is not following this order. It is a rough order. Time to time these may change. So this is the diagrammatically shown view of this lesson. So these are the recombinant vector. It is restriction digestion is occurring. A gene of interest is digested and separated here. It is ligated to the vector, and it is transformed into a bacteria, and also it is cultured using DNA cloning. So that is the overview of our technique. So isolation of DNA from different sources. Different sources can be any organism. Most of the time it may be wild animals as well as humans. Animals mostly taken, and plants also are used as sources and having some important genes like pest resistance. In animals, sometimes protein for dissolving blood clots, and some microorganisms are also used, having some important genes. And when these molecules, recombinant molecules, are inserted into some organisms, transgenic organisms, or useful plants or animals can be formed. So, when our sources are sorted, we are digesting the desired DNA fragments, because in different sources, DNA might be in chromosomes. If it is prokaryotes, it may be in plasmids. Whatever it is, those are longer molecules. We need only a particular gene. We are going to cut it, and also the place where we are going to insert also we have to cut. For that we are using precision enzymes. So these enzymes cut particular gene in particular places. Gene of interest as well as the vector is using the same restriction enzyme, because it should have the same nucleotide order to stick with each other using ligase enzyme. So those cut ends are known as sticky ends. These are sticky ends. Now they are ligated here. So the enzyme is DNA ligase. So DNA fragments are integrated from different sources. DNA using DNA ligase. Usually there should be a cloning site. The cloning site is the site in the vector where the DNA to be cloned is inserted. That is the cloning site. So this is a cloning site. So the cloning site contains sequences which carry sites for many restriction enzymes. So that site is known as a multiple cloning sites, because using different restriction enzymes we can cut it whenever necessary. So usually a host cell is usually a bacteria. It is a bacterial host and can copy the vector, then it can be transformed to the recombinant organism. So this is the separation of DNA fragments by agarose electrophoresis. It is also another technique which we are using. So when we ligate the DNA fragments with the DNA of interest or recombinant DNA, we can retrieve them by harvesting the host cell, or releasing the vectors by lysis, or isolation of the vector using different laboratory techniques, or cutting the DNA which we formed using the same restriction enzymes, and we can isolate it using another set of laboratory techniques. So using any of these methods, recombinant DNA can be isolated, and that can be detected using agarose-gel electrophoresis. In that, we have to use a marker ladder to show their size and to identify. So those are usually these are some. It is like same as the technique we used to isolate DNA, but DNA molecules usually goes down here, since they are small parts or fragments of DNA. It may remain in a particular place. These are DNA marker letters showing some value of the because we can understand the size of the DNA fragment using the marker letter. These are different genes having different sizes. From the marker letter we can get the size of the DNA fragment, and then we can identify the DNA fragment easily. So now we have combined the DNA from different sources and we have identified them. Now we are going to insert the DNA molecules to the host cells, because now we have to form an organism which give a useful product. So we have to insert it. So in that case, we are using the method transformation. There are many methods. The simplest method is transformation, but um, we are using the direct uptake because the cell membrane is having an ability to integrate the DNA. So that ability is used to carry the DNA into that particular cell. So then the host then copy the plasmid carrying the DNA of interest. So this is how we are. This is the recombinant molecule. When we keep it in different chemical conditions, they integrate into the host cell, and now that cell can copy the particular recombinant molecule. So a number of recombinant plasmids is present in each descendant cell of the colony of bacteria cells, because it forms a colony, it copies, and bacteria usually reproduce easily, so it forms a colony. This is a colony. So now we have got a colony of hosts which are having the new recombinant molecules. But the efficiency of the transformation is very low. So here some colonies are not integrated properly. This is properly integrated here, partially integrated here, not integrated at all. So the efficiency of transformation is very low. It means for one transformed host cell, there are millions or billions of cells that are not transferred. So we say the efficiency is very low. So both transformed and untransformed cells will make colonies in a suitable medium, because now bacteria is alive, so they form colonies. We have to identify whether the colony is having the new recombinant molecule or not. So that is called screening. That is also very important. We have to identify the particular recombinant molecule properly. So for in that case, because the efficiency is very low, a sort of a marker should be used. So in that case, we are using antibiotic resistant genes. Then transformed cells can be screened. So usually bacteria is not resistant to bacteria, not resistant to antibiotics. So we are using that property because when we put antibiotic resistant gene, if that bacteria is grown, it grows. Otherwise, usually it is not resistant, so it is a problem. So here the host cells will not grow on a medium containing the antibiotic. When we insert antibiotic resistant genes, that is the marker. So transformed cells can grow in a medium with antibiotic, while untransformed cells are not growing in that particular antibiotic medium. So this is a antigen resistant gene. So when marker gene is used, this is the desired gene and this is the marker gene. So when we grow in the presence of antibiotic, they can grow because they have an antibiotic resistant gene and as well as the desired gene. So because it is coming all the time with the desired gene. So if it is not transferred, both are not coming into the host cell, so cells will not grow. That is how we screen. So there are problems when inserting the DNA molecules through the host cells. It is a difficult step and because cells show some resistance to some foreign molecules. That is very important for for the cells because it can be harmful if it is happening naturally. It is actually happening naturally. When it is naturally occurring, invading DNA can come into a particular organism. So in that case, that is an important property. But in the technology when we use, it is a difficulty for us. So therefore, a large number of copies of the recombinant DNA molecules are necessary to make sure at least few copies are integrated to the host. So we have to produce large number of copies in that case to confirm at least few molecules are getting the recombinant molecule. In that case, we are using in vitro multiplication method known as DNA cloning. So this is up to here. It is we are ligating, we are cutting the DNA and ligating here, and we have formed a recombinant molecule here. So we have transformed it to a bacteria here, and now uh, uh, to confirm it, we have to clone reproducing large number of population of the recombinant molecule. So the DNA cloning always forms an exact copy of a particular organism having the same phenotype as well as the genotype. So here the recombinant molecule again is cloned here, exact copies are formed. So DNA replication machinery of the host cell is used in that case. So that is a normal process. We can use it in this DNA cloning method, but that is done in laboratory conditions. So for a bacterial plasmid to replicate, origin of replication should be there. So origin of replication or ORI sequence is very important. It should be there with the gene of interest. So we have to insert that also in the case of reproducing it under the DNA cloning. So it is combined, and then when it is integrated with the gene of interest, and then when it is go to the host cell, it can reproduce easily. There are many applications in DNA cloning. So it is mainly the study of genetics, genomics and gene expression, and also pharmaceutical research or medicine. And also in medicine there's another area known as gene therapy, which are used as a cure for some genetic diseases, and also recombinant protein productions. These proteins can be used as vaccines as well as many antibiotics, and also cell engineering. We are using forming recombinant cells, and it goes to transgenic organisms. Those cells we are integrating into organisms and forming transgenic organisms. Those are some of the areas which are using DNA cloning. And in DNA cloning, vectors are very important. Any self-replicating unit is known as a vector. It is usually a host cell, usually bacteria, plasmids or bacteriophages. It means bacteria viruses are used in as vectors in cloning, and DNA molecule of interest can be integrated into these vectors. They have plasmid DNA, and it can be integrated into these vectors. And vectors are the vectors to carry DNA of interest into the host for multiplication or cloning. So there are many types of vectors. These are some of the types: plasmid vectors, phosphid vectors, and also some recombinant vectors are also there. And there are many types of vectors. In our lesson, recombinant vectors are very important. When the vector is carrying foreign DNA, it means a recombinant DNA. When it is there in a vector, it is a recombinant vector easily. So this is the host plasmid, and it is cut here, and then some new genes are inserted here. So it is a recombinant vector. And there are cloning vectors also. vectors which we are using in DNA cloning are known as cloning vectors, and they are copied in an in vivo system. This is inside the living cell. Though we do it in a laboratory, it is happening inside the living cell. So it is in vivo system. So for this, the number of copies in a single host has to be very high, and this condition is also fulfilled by bacteria, bacterial plasmid, bacteriophages, and there are some yeast artificial chromosomes or X. This is a normal common cloning vector, and cloning vector also should have organ of replication, multiple coincides and markers. Otherwise, a vector cannot be used in DNA cloning. So, yeast artificial chromosomes or vectors. So, plasmids are present in yeast cells also, like in bacteria, and those are known as yeast artificial chromosomes or YACs, because they can work as chromosomes when they are linearized. We can cut at one end, we can linearize it. They can act as chromosomes because they have some sequences related to centromeres. Centromeres are in usually chromosomes, as in eukaryotic cells. So when we are linearizing, they can, because they have those sequences, they can act as a chromosome. So that's why, though it is a plasmid, we are known it as a yeast artificial chromosome. And they also have autonomously replicating sequences or ARS. So they have some ARS3 sequences. It means other than cell division, they can replicate inside an organism. They have some sequences like that. Those are very important features in the yeast artificial chromosomes, and it is fulfilling all the criteria for a vector, so we can use it easily as a vector. So there are advantages of YAC vectors. They are very large. So large amount of DNA can be copied using them, and also they can operate in an eukaryotic system. So usually as a host to vectors, we are using bacteria, bacteriophages and all. Since they are yeast artificial chromosome, they can act as chromosome, they can work in eukaryotic systems. So there are some DNA delivery systems for recombinant molecules to introduce into a host. We discussed about transformation, the most easiest method, and there are, other than that, transduction, gene gun, and agrobacterium mediated gene transfer is used as other methods of DNA delivery. Transformation, we discussed already. A large number of copies of DNA of interest is mixed with the host cell. So, ability of the cell membrane to integrate into a cell is used here. When we mix and keep the necessary conditions, it is integrated into a bacteria. So, but here the efficiency is very low of transformation. Various treatments are taken to increase the efficiency. Transduction is another method. We are using the bacteriophages here. Bacteriophages means bacteria viruses. This is a bacteria virus, bacteria infecting virus. So that bacteriophage is having a property. It can insert its genome, it may be DNA or RNA, into the bacteria, and it can using the machinery of the bacteria, it produces its necessary DNA for the bacteriophage. So that mechanism is used here. So if we can integrate some DNA of interest into this bacteriophage genome, it will go easily to a host cell. So that is the method used here. So gene of interest, it is integrated into the modified viral genome and packaged into the protein capsid, and protein capsid also protects that DNA. So viral particle can pass the recombination molecule using its normal infectious process. The capsid protects the DNA, so this method is more efficient than transformation. This is an more advanced method, the gene gun method. This is the gene gun, it is like a gun, and it is using some small particles of heavy metals such as gold. And those small particles are coated with, here the particle, it is coated with large number of copies of DNA of interest. So these particles are short and high velocity. Here a piston is there, using that a high pressure is formed and it is shot to the, this is some tissue, maybe some tissue. So it is inserted into that foreign tissue easily with the pressure. So gene gun is used here. This is one of the most advanced techniques in gene transfer. And there's another method, agrobacterium mediated gene transfer. This is also a natural process. Agrobacterium is a soil bacterium. It can infect plants. When it infects plants, it forms a disease. It is the crown gall disease. Actually, crown gall disease is a tumor in the plant. A tumor means the bacterium is living inside it, and the cells of the plant are genetically transformed by the bacteria by a segment of agrobacterium. So, if we can give our needed DNA to this bacterium, it is usually transforming the cells of the plant. So, we are using this method. The part of the plasmid can be transformed to the necessary DNA, and this DNA is known as tDNA, and plasmid is known as tumor inducing plasmid. So whatever it is we are doing, actually for the bacteria we are giving the gene of interest and it transforms the necessary product to the plant. Here, when we do this technique, we are to insert that particular plant DNA segment to the bacteria. We are using a method. We are actually get away the disease causing part of the bacteria. So formation is of this tumor is not occurring. So what we do is right and left border of the sequence of tDNA is needed, and most of the needed bacterial genes are removed here because we need space to insert the gene of interest. So when we remove some genes, some virulent genes are also removed. It means this is causing genes. So what happens there is, then agrobacterium can deliver this tDNA using its infectious ability, infecting ability, and it new plant is not getting the disease, but the new DNA molecule is getting to the plant. So this is also one of the most widely used technique because it is a natural process. So that is the end of the lesson. And this is my question. Write short notes, and on the following we come to DNA technology, vectors, and DNA delivery systems. So please answer the question, and thank you for listening, and God bless you.